Dr. Michael Hudgens is a professor and chair of the Department of Biostatistics at UNC Chapel Hill. He also serves as the director of the Biostatistics core of the UNC Center for AIDS Research. He has experience in collaborative research and statistical methodology development related to the studies of infectious diseases. Without further ado, Dr. Hudgens, please.
Hi, thanks everyone, and thanks for the invitation to be here. It's very nice to be here. Although I guess you all in North Carolina, we have this thing called spring, like sunshine and flowers and such. It's really novel. You might enjoy it. But otherwise, it's great to be here. Thanks for the invitation. So yeah, I'm going to talk for 35 minutes or so, I've been told. Mostly 90% of this will be about sort of spillover effects in vaccine studies. So our group in general is interested in the development and application of causal inference methods to infectious disease studies, which I think is near and dear to the heart of many of you all. We think about both trials and observational studies. We mostly focus on preventative studies and less on treatment trials or treatment studies. And there are sort of two problems that I've been interested in, particularly over the course of my career. One is assessing immune correlates or surrogates of protection in vaccine studies. That's a problem I've worked on quite a bit over the years with Peter Gilbert and others at the Fred Hutch Seattle. And then the other problem, which is the one I'll talk to you about today, is assessing vaccine effects in the presence of potential spillovers, or what in the causal inference lingo we refer to as interference. So drawing inference about effects of vaccines or other preventive measures in the presence of interference. A couple of other problems that we've worked on sort of in this space. One is the post-infection selection bias problem that occurs when you do preventative trials, and then you want to look only at a subset of folks who get infected. And then also dabble a little bit with generalizability or transportability problem, which I know Lisa and others here at Harvard have worked on quite a bit. But for today, I'm going to focus on this, mostly this interference problem. So typically in causal inference, we start by thinking of individuals as having two potential outcomes. Apologies, I'm using X here for exposure, not A in the Harvard way. So we usually think Y naught and Y1, individuals have two potential outcomes. And then we ask questions like what happens on average if everyone's treated, what happens on average if no one's treated. But what if individuals interact or connected in some sort of way? And in particular, what happens if the treatment of one individual potentially affects the outcome of another individual? How do we extend sort of classic causal methods to deal with that problem? And that's what some people refer to as interference, that whether or not I'm vaccinated might affect the outcome of somebody else, including my partner. Most of causal inference methods assume no interference, but it's clearly not true in some settings. Clearly, there's interference that will be present in some settings, and infectious disease is sort of the leading example of that motivates this work. Lots of other settings where you can imagine interference might be present. For example, in educational settings, you might think that there might be interference between two students in the same classroom. Or in agricultural studies, there might be interference between two plants that are vegetable sharing. In some settings, interference might be a nuisance, something you kind of want to design away. But in other settings, and in particular in an infectious disease setting, this is something we really need to study and understand, a phenomenon of interest. There are lots of other areas where there might be interference or spillover effects. Sometimes these are called pure effects as well. So sort of language, the nomenclature varies across literature. But there's been work in a variety of fields that I'm listing here. In the interest of time, I'll just skip over this. But suffice it to say, these sort of interference, spillover, or pure effects are of interest in many different substantive areas. And for that reason, there's sort of methodology coming out from different folks. It makes it a really interesting area to look at. Here are a few other areas where you can see these types of methods being developed and deployed. Okay, so I'm going to sort of talk about methods broadly that fall into one of 4 categories. So I'm going to think about scenarios where we have clusters first, and then talk about when we don't have clusters and instead you just have a network. And then think about first when we have randomized studies, because causal inference is easier when we have a randomized study, and then think about observational studies. So we'll start with the easiest possible problem, which is like you have clusters of individuals and you can do a randomized experiment. And in the literature, people refer to when you have clusters of individuals and you're willing to assume that there's no interference between individuals in different clusters as a partial interference assumption. So suppose you have clusters, suppose you're willing to assume that there's no interference between the clusters. And then the goal would be to draw inference about treatment effect that allows for, doesn't insist upon, but allows for the potential for interference between individuals within clusters. So these types of methods would be appropriate if you had, for example, students in schools or people that were in villages or some clear way to put individuals into clusters. One of the things that makes the interference problem interesting is there are a lot of different ways to define causal estimates. Catherine and I have been talking this morning about different ways she's thinking about finding causal estimates in the presence of interference. I'm going to sort of just paint with a really broad brush at a high level what the type of estimate you might think about without getting into the details. But I'm going to be thinking of policies which correspond to counterfactual scenarios where a certain proportion of individuals in the population get treated. So note that by alpha. So when you say alpha as a policy, just think alpha percent of the people are getting treated. And then you can think of average outcomes under that policy. Okay, so mu alpha would just be the average potential outcome under the policy. And we can also think of the average outcomes depending on whether or not an individual is treated. So mu x alpha would be the expected or average potential outcome when an individual receives treatment X, and policy alpha has been deployed in the population. And then you can define treatment effects or vaccine effects as contrasts in these average potential outcomes. So for example, we might define an indirect effect or a spillover effect as mu naught alpha naught minus mu naught alpha 1. So that says what happens on average when you're not vaccinated and you change the proportion of people around you that are vaccinated. So if there is interference, if there is a spillover effect, then that would be non-zero. We know in causal inference, if you do a randomized experiment, drawing causal inference is straightforward. So you might think in this setting, what would be kind of the ideal randomized experiment to do? So one idea would be to do a 2-stage randomized experiment, where in the first stage, you randomize clusters, different policies, different alpha values. And then in the second stage, randomize individuals to treatment or not, depending on the policy assigned to a group in the first stage. So this is an example of such a study that took place in Malawi, where the clusters were these so-called enumeration areas. So these are like census districts. Each of these enumeration areas was assigned to one of 4 policies, alpha 0, a third, 2/3, or 1. And then in the second stage, the individuals who participated in the study, who were women who are not married, ages 13 to 22, they were randomly assigned to get the treatment or not. The treatment here was a cash transfer. But the likelihood that they got assigned treatment depended on the alpha that was assigned to their cluster in the first stage. So there have been lots of methods written about drawing inference about different treatment effects. When you do this 2-stage randomized experiment, get some references here for you. I'm not going to go into this today, but some of these methods use randomization-based inference, others use large sample frequency inference. In either case, it's fairly straightforward. And the reason it's straightforward is because you've done this 2-stage randomized experiment, you don't have to worry about confounding either at the group level or at the individual level. And so because of that, and because of the fact that we have clusters of individuals and we made this partial interference assumption, generally inference is straightforward if you're able to do these sort of two-stage randomized experiments. All right, so suppose next that we have an observational study, so we still have clusters, but we no longer have the luxury of doing randomized experiment. What might we do then? And there's been a lot of work on this in the last 10 or 12 years. And basically what people have done is extended what are sort of called classic SUFA methods, or methods that assume no interference, extended those to allow for partial interference. Good. And these methods, this sort of class of methods that have been developed, generally rely on two key assumptions. One is sort of envisioning that the set of clusters you have is a random sample from a superpopulation of clusters. And then the second is some version of the no unmeasured confounders assumption or the exchangeability assumption. But it's a no measured confounders assumption at the cluster level. So assuming that the cluster potential outcomes are independent of treatment assignment for that cluster, conditional on the right set of covariates. So for example, Eric and Tyler, this paper in 2012, we're all friends. I can say Eric and Tyler here, right? Everybody knows what I mean. So they had this paper in 2012 where they suggested these IPW type estimators, the extensions of the classic IPW estimators to allow for clustered or partial interference. And then this 2014 paper, Perez Hedrick was a postdoc in our group. She took these IPW estimators that Eric and Tyler proposed and derived large sample properties, then applied them to a vaccine, observational vaccine study data that I'm going to show you in a minute. And so we spent quite a bit of time looking at and working on these basically the original IPW estimator that Tyler and Eric proposed. Have a doubly robust estimator that was proposed in 2019, and then I'm going to show you some results from this paper in 2022 by Chakwadar et al., where we basically extended to allow for censored outcomes by including inverse probability censoring weights as well. So the extension to deal with the censoring weights is not particularly exciting. It's more, I just want to show you sort of these methods in application to real data. So the real data comes from a cholera vaccine study that took place in Malab, Bangladesh. This was actually an RCT, so it was an individually randomized trial, but they collected data not just on the participants, but on a larger group of individuals in the area at the time. So it has an observational component as well. And so if you're going to analyze the data from everybody, you need to account for that. And there have been previous analyses of these data based on sort of the spatial distribution of individuals and where they live, and also using social network information about who in the study knew each other to suggest that there's potentially a spillover effect or interference here. So analyses that showed amongst individuals who did not receive the cholera vaccine that their risk seemed to go down as the proportion of people around them vaccinated went up. So there's some evidence, some hint in the earlier analyses that there was an interference or spillover effect of the vaccine. So in this analysis, I'm going to show you involves about 94,000 individuals partitioned into 700 clusters or groups based on the spatial distribution of where they lived. The outcome of interest is time until cholera, so it's like a survival time, subject to right censoring due to dropout, emigration, and end of the study. And then on the next plot, I'm going to show you the results of applying this sort of IPW type estimator, where we're estimating the cumulative risk of cholera under three different counterfactual scenarios. One where the level of vaccine coverage in the population is 30%, then 45%, and 60%. So these are the results. On each of these curves, the black line is showing you the cumulative risk when you're in the scenario where you're not vaccinated. And so you can see as you go across the plots, that cumulative risk curve just plummeting. And so that's the spillover effect or the interference effect. If there was no interference, you would not expect the risk when unvaccinated to have anything to do with the level of vaccine coverage in the population. So we see a pretty dramatic spillover effect if you're unvaccinated. In contrast, the blue curves here showing cumulative risk if you're vaccinated, they tend not to change as much across the three plots, suggesting that a sort of spillover effect is less pronounced, if at all present, when vaccinated. So basically, if you're unvaccinated, it really matters what the proportion of people around you vaccinated is. If you're vaccinated, it doesn't seem to matter so much. So I mentioned earlier, one of the things that's interesting to think about is what are the causal estimates? What are the parameters we're trying to draw inference about? And these first set of papers that came out on observational data with partial interference targeted the same estimates that were proposed from the papers that thought about the two-stage randomized trials. And so basically the estimates here are considering counterfactual scenarios where individuals independently select treatment with probability alpha. Tyler and Eric in their paper, they call this type B, B being Bernoulli, right? So we go around and we flip a coin for each person, comes up heads with probability alpha, and you get treatment if it comes up heads. So that's fine in the RCT setting, but in an observational setting, I would submit it's not clear that this is necessarily the most interesting counterfactual scenario to think about. So if we're in a world where we have clusters and we're thinking there might be interference within clusters, there's probably dependence within clusters. And in particular, there might be dependence in the likelihood individuals select treatment within a cluster. And if that sort of dependence exists in the factual world, then we might expect that to also exist in some counterfactual world. And, being in the school of public health, we think about, well, what if we could get more people vaccinated, right? And so we may live in a world where there's some sort of dependence in the propensity scores amongst people in a cluster. And even if we could encourage more people to get vaccinated, we might still expect in that counterfactual scenario some dependence between treatment selection amongst individuals in the same cluster. So we've been more recently focused on other estimates, which we think are more relevant from a public health standpoint or a policy relevant from a policy perspective. And the basic idea is to think about sort of a propensity score distribution and trying to shift it. And so not insisting that everybody gets treatment with probability alpha. Some people are still going to be less likely to get treatment, others more likely, but on average, propensity score is roughly alpha. So that's the idea. I've got a couple of pictures here to try to explain to you, give some intuition about what's been done in the past and what we're trying to do now. So imagine you have a cluster with two individuals, and we're on this blue dot. Okay, so individual one has a propensity score that's about, I think, 0.65, and individual two has a propensity score about 0.15. So their average propensity score is 0.4. Okay? And in fact, anywhere on this diagonal line, the black diagonal line, the average propensity score, any pair of points on that line, the average propensity score will be 0.4. Okay. All right. So imagine that's where we live in. And then we say, okay, what if we lived in a world where instead we're up on this red line? So the red line, the average propensity score, I think it's about 0.75. Okay. So any pair of points on that line will correspond to a pair of propensity scores that average to 0.75. So what the type B policy estimates were doing and those sets of methods were saying, what would happen if we moved to this triangle here? So we moved from a scenario where individual one was more likely than individual two to get treatment to a scenario where they're both equally likely to get treatment. So what we're sort of saying is that in many settings that may not really be the relevant counterfactual to think about. And instead, we might want to be somewhere down here in this region where that ordering between the individuals stays the same. So if in the factual world, I'm more likely to get vaccinated than Mark, then in the counterfactual world, I'm still more likely to get vaccinated than Marcus. So one idea would be to kind of pick the closest place on the red line to the factual world, although then it's sort of unclear where you might map some of the other pairs of points. So we're actually taking the approach on the right. This curved line segment is the place where the odds ratio between the two individuals is the same. So we're going to maintain my odds of getting vaccinated versus Mark's and pick the place in this counterfactual scenario where the odds ratio is preserved. So that's the idea in this paper by Barkley et al. And Papadoe Giorgio had basically the same idea in the paper that came out right around the same time. Ed Kennedy also has a paper that talks about this idea of kind of shifting everybody's propensity score the same amount on the odds scale. That came out in JASA a few years ago. That's Kennedy's papers in the absence of interference. But all three of these papers are sort of thinking about the same idea. Instead of the classic causal estimands of what if we treat everyone compared to what if we treat no one, what if instead we sort of shift the propensity score distribution, arguing that in many settings that may be a more interesting causal estimand to try to draw inference about. These are the results from the Barkley paper. So reanalyzing that cholera vaccine trial data, but instead of targeting this type B policy estimand, instead looking at this incremental propensity score type estimand. The upshot of these results is that it's the very similar sort of take-home message. What we're looking at here are spillover effects. The blue lines and points correspond to if you're unvaccinated. You can see that the spillover effect estimates, the sort of slope is greater for the unvaccinated group compared to the pink lines. Those correspond to point estimates and confidence intervals if you're vaccinated. So it's the same take-home message, which is the spillover effect is stronger in the unvaccinated compared to the vaccinated. We just have a paper that's coming out now in JASA that basically all of the methods I've showed you rely on some sort of parametric model. Tyler and Eric's original paper had a parametric propensity score model. The doubly robust paper that I sort of passed by earlier, it's doubly robust, but you either have to get a parametric propensity score model right or a parametric outcome regression model right. So in this paper, we do everything non-parametrically and sort of work out if instead of estimating the propensity score using a parametric model, I use machine learning type methods, and then I do the same thing with an outcome regression model, and I put them together and come up with a non-parametric way to get at these same types of estimands in the presence of partial or clustered interference. Originally in this paper, we targeted the incremental propensity score type estimand, but in the process of revising it for JASA, we generalized it. And actually, the results now hold for any policy. So any policy that you can think of where you want to modify the propensity score distribution is covered by the methods in this paper. So we look at not just the type B and the incremental propensity score, we also look at one where you truncate the propensity score distribution. So you insist that no matter what, every cluster has at least a certain percentage of people treated. We look at another counterfactual policy where you shift the propensity score distribution only in a subset of clusters, maybe clusters that are particularly high risk. But the methods are set up quite generally to handle any sort of allocation policy. All right, so now let's suppose that instead of having clusters, I give you a network. So we've sort of been thinking about the picture on the left, where you can take individuals and put them into clusters, and you might allow interference within clusters, but we were insisting no interference across clusters. So suppose instead we have a problem that you just are given a single network. Okay. I'll start, I'm only going to talk about the simpler problem, which is you have a network, but fortunately we've been able to do randomization. This is data from a RCT that was done about 10 years ago by Alison Aiello. She's a professor of epi at Columbia. She was at UNC. let's see, this study took place in a large university in the Midwest. The exposure or treatment was to self-isolate in your dorm room when you developed influenza-like illness symptoms. And then the outcome was looking at IOI in the participants. The left graph shows you the sort of snowball type of sampling that was used to enroll participants in the RCT, with the red nodes indicating seed participants. And then if you were in the RCT, you could invite one of your friends to participate in the RCT. And then your friend could invite someone and so on. So you get this chain referral type process. And we might think, since people are referring each other to the RCT, there might be interference between individuals who refer each other. On the right, showing the edges between nodes here indicate individuals have identified each other as friends. So I know somebody else in the trial. And these individuals were in residence halls. So there's multiple reasons why we might think that there's interference between individuals in this RCT. So I'm going to show you a really simple method for drawing inference about treatment effects.
I think it's for you.
All right, so I'm going to show you a really simple approach to drawing inference about treatment effects when you have network interference, but you have the benefit of doing randomization. And this is from a pair of papers by Bowers et al. And then I'll show you an application of it where we extend it to deal with censoring. But the basic idea is really simple. So I'm going to assume that we know something about the interference structure and that we encode it in a matrix of zeros or ones, where 0 in the matrix indicates that I'm willing to assume there's no interference between two individuals. Okay, so Aij is 0 if I'm willing to assume individual I and J don't interfere with each other. Otherwise, I put a 1 in the matrix. And that'll be our A matrix. And if you're willing to assume partial interference, then you could sort the rows and columns of that matrix so that we block diagonal. But otherwise, we're going to let it take this general structure because we're thinking about networks now. And this is the idea of the Bowers et al. Approach, just 2 slides sketching it out. So imagine for each person, we do the randomized study. So we observe the treatment assignment, that's X. We observe the outcome Y. I'm going to use this bold X to collect up the vector of treatment assignments for everybody. And then write the potential outcomes for each person as indexed by that entire vector. So I'm allowing for general or network interference. So in the most general setting, then this treatment assignment vector, it's of length n, takes zeros and ones, so it takes on 2 to the n possible values. So each person has 2 to the n potential outcomes. It's way worse than the usual causal setting where everybody just has 2 outcomes, 2 potential outcomes. One of the potential outcomes of interest that we'll focus on is why not. Rosenbaum calls this the uniformity trial. So this is the potential outcome if no one got treatment. And then the way the Bowers et al. Approach works is you posit a causal or counterfactual model that allows you to relate the observed outcome to the uniformity trial potential outcome. And this causal model can take different forms. I'm going to show you one simple one, but there are many different ones that are considered in the couple of papers by Bowers et al. I like this one because it has a simple accelerated failure time model type of form that people are familiar with. So this just says that the observed outcome is equal to the uniformity trial potential outcome times this piece here. Xi that's the treatment assignment for individual I. So the coefficient on that will have an interpretation as like a direct or individual effect of treatment. And then GI, which is defined here below, is just the proportion of individual I's neighbors that were treated. And so the coefficient on G will give us some indication of the spillover or interference effect. Okay. So you posit this sort of model, and then essentially because we have randomization, and I'm skipping over all the details here in the interest of time, but you can invert a randomization test to construct point estimates and confidence intervals for delta and T. And I'll just show you what these results would look like if you applied them to the vaccine study I mentioned earlier. For this analysis, I'm just going to subset to the actual participants in the RCT because this Bowers et al. Method is only designed for randomized experiments. So I'm ignoring the data that we have beyond the RCT. And these results from this paper by Wenwei Lo, who's a postdoc in our group, basically extends this approach to allow for right-censored outcomes. I'm going to show you three different results. This method requires you to make some assumption ahead of time about the interference structure. So we want to know how robust were the results to that assumption. So three different interference structures are assumed. One is that we could put individuals into clusters based on where they live, which is essentially the type of assumption and that we made about the interference structure in the analyses I showed you earlier. But now we're also going to allow for more interference structures that do not require clusters. So one of them is this 500-meter interference structure where basically each person, you draw a circle around them of 500 with a radius of 500 meters and allow for individuals within that circle to interfere with them. And then the other is based on a social network information that's available. So these are what the interference matrices look like for those three different assumptions. And these are large matrices, 73,000 by 73,000. It's a little hard to see what's going on. It actually looks like the social network matrix is the densest, but if you zoom in, I think you can see it's actually not the case. So this is showing you a 500 by 500 submatrix of those larger matrices. And then these are the types of results you get. So you essentially invert this randomization test and you can construct a contour plot of the p-values. And so that p-values contour where the p is 0.05, the interior of that gives you a 95% confidence set for the parameters, delta being the direct or individual effect and tau being the interference or spillover effect. And then the point estimate here is the place where that p-value is the largest. So in any event, regardless of the interference structure assumed, we see evidence of a significant direct and indirect or spillover effect of the cholera vaccine. The model I showed you, I think, is nice because it's easier to interpret the results. We're running short on time, but just suffice to say, it's very straightforward, I think, to interpret the point estimates that come out of this analysis. And then I think one of the other papers that maybe some of you all read was this paper by Shana, Alexandria, and Allison, where we applied the same sort of method, essentially the Bowers et al. Method, to that study I showed you on social isolation. And it's basically the same idea, but it was combined with kind of a transmission probability model. You might say, well, that's great, but I didn't like that part where you had to assume a causal model. I really don't like modeling around. I didn't like the fact that you had to make some assumptions a priori about the interference structure. So is there anything you could do that would be a little lighter on the assumptions? So there has been some work that just tries to detect whether or not interference is present. So it's completely non-parametric, but there are this series of papers that develop tests for seeing is there any evidence of a spillover effect at all. We have also this paper, it's published a couple of years ago, where we said suppose you're a randomized study and you did something simple like difference in sample means or difference in Horwitz-Thompson type estimators. Would you still get, in some sense, the right answer as the effect, as an estimate of the direct effect of treatment, even if interference was present? And the answer in this paper is surprisingly yes. You can still sort of interpret your results as estimating a direct effect, even in the presence of interference in a randomized experiment. And what's nice about this paper is that we really don't assume you know much of anything about the interference structure, except maybe some summary statistics, like the number of ones in that interference matrix. But that's it. We make no other assumptions about the interference structure. So the last cell, the 2x2 table, this is the hardest problem. So we do observational data on a network. This is hard because you don't have the benefit of randomization, so you can't leverage that. And you also don't have independent replicates. You don't have clusters or groups, so you can't leverage that either. So that's a much harder problem. You'll need some sort of inferential method that adjusts for confounding and allows for dependent observations. And there has been some work done on this. So that's the end of my interference part of my talk. It's a really exciting area, lots of interesting problems. Most of them come out of, in my opinion, the infectious disease vaccine studies, but lots of other areas as well. And then I just want to close by mentioning a non-interference paper that we just put up on archive. And I would really be interested to hear from you all if we're kind of reinventing the wheel or what we've done here is new. But in this paper, we do a nested trial emulation to try to assess the effect of COVID vaccinations using observational data, relatively large data set from Italy in 2021. And what we wanted to know when we looked at these data was whether or not the vaccine effectiveness was changing over time. And we were interested in two different timescales. We really wanted to know in both calendar time, so is vaccine efficacy going down because of new variants, and the other time scale being time since you got vaccinated. And I'll show you the results in the next slide. What we found was that it looked like vaccine efficacy peaked around 15 to 20 weeks after the first vaccination, which seemed to us to be consistent with what we would expect. And then also some evidence perhaps of a modest decline in vaccine efficacy over calendar time. And this work does not allow for interference, but I think a natural question would be if you had information, say, about where individuals lived, could you extend this nested trial emulation approach to allow for interference? So these are results that we got. So this is the same plot from two different angles. The solid curve is the point estimate of the vaccine effectiveness. This is on the usual 1 minus relative risk scale. And then the transparent surfaces are pointwise confidence intervals. And basically with this shape, this kind of curve that you see, that's the vaccine efficacy peaking about 15 to 20 weeks after the first vaccination. And then I guess, let's see, on the left plot, as you see that it decreases somewhat from left to right, that's a decrease in vaccine effectiveness over calendar time. So that's it.
What are the years? What are the dates that papers can't be?
This is all in 2021. And so it's a series of nested trials. So we're imagining that we did a sequence of randomized trials, one per week, starting in February into May, and then ran them till December of 2021. So that's it. Just been fortunate to work with lots of great folks over the years, and to receive funding from the Gilling School of Global Public Health at UNC, as well as NIH. Thank you all again for the invitation to be here and your attention today, and happy to answer any questions. Mark, you have a question about this slide?
No, I need to think about that one for longer. I did have a question about there was a slide where you had the pink and purple, and it looked as though some of the spillover effects went negative.
Yeah. So this is comparing, right? So I'm computing these spillover effects as defined here. So if X is 0, then it's the spillover effect if you're not vaccinated. So basically saying what happens if you're vaccinated and the coverage is alpha compared to alpha prime, right? So on the left, alpha prime is 0.3. And let's see, so this bar, I don't know if you can see my cursor.
Yeah.
So this bar point is saying, what's your risk if you're not vaccinated and coverage is 20%, compared to 30%. Okay, so positive numbers here on the far left
I see.
would indicate you're at higher risk than had coverage been 30% of the population. And then as you go this direction, they're negative because that means you're at lower risk relative to had the coverage been 30%. Yeah.
So it's not the spillover effect of more vaccination is negative, it's the group where.
It's anchored at 30% in that plot, and the right one's anchored at 50%.
Thanks for a super interesting talk. This question is a little bit informed, but would love your thoughts on it. Most of the examples that you were talking about, particularly around the cholera vaccine, seem to me like if you were doing a naive analysis at the cluster level, you would find a larger effect than you would if there was no spillover effect. The spillover effects were positive. And so from a policy perspective, it seems like that is a case where like what you would see with the naive analysis looks good, looks better because of spillover, and you'd be more inclined to do a policy or to implement a policy. On the other hand, in the cases where you don't have clustering in that, which the clusters that prevent interference between treatment and comparison, I could imagine that the naive effect that you would get from doing some kind of clustered analysis would be smaller than otherwise expected because you have sort of mixing between treatment and comparison groups. And I was curious how you've thought about the implications for that case for policymaking, especially when there's a lot of uncertainty around what those connections might look like.
No, it's a great question. I do think largely a take-home message from all of this work is that you can be misled if interference is present and you ignore it. I referenced that one paper by Frederick Save that said there was an exception to that. If you do a randomized experiment and you're targeting the direct effect, you can still sort of get the right answer. But otherwise, you can be misled. And I don't think there's a simple way to describe that you will largely be misled in the following way. I think it depends on the scenario. One thing that probably doesn't come across in this set of slides, but I think it's important to think about things like the direct effect or spillover effect, to think of them no longer as a single number in that case. There's not a direct effect when there's spillover, but rather think of it as a function. So there's the direct effect is a function of alpha. It's a function of the level of coverage. And so, the direct effect of receiving a vaccine could be huge if you're in a setting where no one else is vaccinated. But if you're in a setting where almost everyone else is vaccinated, the direct effect of receiving the vaccine is probably very small and minuscule. And so I think those are the ways we can be misled, right? So if you do a study and you want to understand vaccine effectiveness and you happen to do it in a population that's already, there's decent coverage, you might come to the conclusion the vaccine has no effect. In fact, it does. You're just in a situation where there's high levels of coverage.
I had a question about the last set of slides you showed on the nested emulation. I just want to clarify. Here by nesting, you're just using the sequential daily, the nesting part is the amount of time. You're not incorporating any of the group level components.
Right. So no groups here. We didn't know where these people lived in Italy. We wanted to do something that allowed for interference, but we didn't have that information. So it's nested only in the sense we're imagining that we did a sequence of randomized trials, and when we construct the analytic cohort, that rows in the trial, an individual is allowed to enroll in multiple of those.
And then do you know what the range of alpha is over this period? Do you ramp up, does vaccination coverage in the area ramp up to?
I don't know.
Okay.
I don't know. I think it was very, very low, right? In the beginning of 2021, I think, only the old folks were eligible and frontline workers. And I don't know by December 2021 what the sort of saturation was in this region of Italy. I'm very interested. The data just wasn't available to us.
The other obvious thing about this, which I was just trying to think through, is that your vaccination pretty much all happens before Delta comes along. And then most of the things that are happening in the second half of the year are going to Delta. And I've been trying to put my head around thinking what I would expect from that given that, as you say, the types of people who are getting vaccinated in May, some are different from the types of people getting vaccinated in January.
Yeah.
But yeah, I haven't wrapped my head around that. I just want to point out that our period is almost all delta.
Thanks. I'm curious, I think there's been a lot of trial emulation papers written from this group in Harvard, but have people seen it with two separate timescales? We hadn't seen it before, and that's part of what we thought was interesting. That we could think about both calendar time and time since vaccination.
I don't know of any that really do a ton dependent at all because of the selection problem. That you're not really, that part is sort of it's the spirit of trial emulation because you're conditioning on not being infected at the time. Is this like a hazard ratio or something?
No, it's 1 minus a cumulative risk ratio. So basically our premise was, okay, I'd like to know if vaccine is waning. And I mean that both in calendar time and time since vaccination. And so if resources were not a problem, the way I would do that is I would do a series of vaccine trials.
Yeah.
And then I could answer both of those questions. And so this is a attempt to emulate that.
I don't think that's been done. I haven't even.
Yeah.
I need to think what that means, but it sounds really cool. In the one with the positive model, yeah, that one. There you didn't have an interaction to allow the direct effect to vary with the
That's right.
coverage.
Yeah, so we fit this really simple model.
Yeah.
And you could think of lots of other possible models. I think in practice, probably what I would think we might want to do is fit multiple models and see how our results change as we change the specification of that. Certainly, one to explore would be one with an interaction. What's interesting is that, or one interesting aspect I did not mention is you can get scenarios where if the model is misspecified, which is of course what we worry about whenever we make these modeling assumptions, then it can be the case that you get you get all p-values less than 0.05. So no matter what values of the parameters you run this test on, you reject. And that's a rejection of the model. So that tells you you've selected the wrong model. So that's kind of a nice feature. On the other hand, that means if your model's misspecified, but only a little bit, you can get a really narrow confidence set. And so while you think that might be due to precision, it's actually due to some mild model specification. So the way I like to do this is, this set of parameter values inside the 0.05 contour, the data are compatible with that model and those parameters. And the data very much could be compatible with some other model and some set of parameter values associated with that.
I think that's all we have time for. So thank you so much, Dr. Hudgens. We'll be in this meeting if you have any questions today.